cell signaling or cell communication. Step 2, transduction. That means converting proteins from the inactive state to active form along the cascade. It contains multiple steps. The point of the multiple steps is to amplify the signal as well as make it easier to control and regulate cell communication and response. We have a protein, and besides the carboxyl and the nitrate, the amino group, not the nitrate, amino group, we have a couple of them that contains a hydroxyl group. It will, in the presence of ATP, the enzyme, the protein kinase, will phosphorylase this inactive protein, and it's going to turn into an active protein. So the hydroxyl group will bind with a phosphate group, so an active protein, and ADP is the side product. One was inactive, and now we have an active protein through the shape change. To inactivate this active protein, dephosphorylation can take place by the enzyme that is called protein phosphatase. So, and also a phosphate, inorganic phosphate will get off from this reaction. So we have the relay molecule that activates the inactive protein kinase, and what can really activate the inactive protein kinase are the calcium ions or cyclic AMP. Cyclic AMP will be a different topic. So it's going to be a cascade reaction because this active protein is going to activate another protein kinase, and this active protein kinase will activate another protein kinase until the target protein is going to get activated. It's a cascade, many steps to amplify the signal as well as to make it easier to control. The other type of transduction happens without proteins at the beginning, but it happens through secondary messenger. These second messengers are small non-protein molecules. They can be the calcium ions and cyclic AMP. Here comes the cyclic AMP. Cyclic AMP, you might just wonder why on earth is that? Well, because it has a circle shape next to the ribose. Do you have to know this formula? No. But for those people who would want to see why it's cyclic. So this happens with the GPCR, the G protein coupled receptor. The ligand, the first messenger, for example, the epinephrine, is going to bind to the GPCR. The G protein is inactive because it has the GTP, but as the ligand binds to the receptor, it's going to move off and it's going to activate the adenylyl cyclase that is going to make cyclic AMP from the ATP. This cyclic AMP as a second messenger is going to activate the inactive protein kinase A to an active protein kinase A. And the active protein kinase A is going to phosphorylate other proteins. So next one, the last, it goes back, this cyclic AMP can go back to ATP by phosphodiesterase enzyme. That it's kind of a dehydration reaction. Okay, response, that's the third step. And we have the cell. And we have the nucleus, the DNA in the nucleus, the cytosol, and the nucleus. So the transcription factor, the final activated molecule, is going to let the transcription and translation happen. So we are going to gain a protein eventually. And also regulates protein activity, how the protein is going to be active or non-active, and also affects metabolic activities like the epinephrine through cyclic AMP, activates enzymes that would break down glycogen to glucose. And this is really good because you will have energy right away.